albeit virtually. And yeah, thanks Ian for joining in the evening in Sydney. So Ian is Professor of Biostatistics and Director of the Biostatistics Group in the Clinical Trial Centre at the University of Sydney, and has previously worked at Pfizer as well as Harvard University. He has a long-standing interest in developing statistical methodology for the design and analysis of clinical trials and epidemiological studies, as well as extensive experience of applying such methodology in practice. So I'm very much looking forward to this talk about estimating treatment effects in adaptive trials. And Ian, the floor is yours.
Thanks very much for the introduction, David. And thanks very much for the invitation. It's very nice to see that we're working on some similar areas of methodological research, and I hope we can use this as a motivation to have further interactions in the future as well. Just by way of background, as David mentioned, I'm from the NHMRC Clinical Trials Centre at Sydney University. We have a group of biostatisticians working on trials and methodological research. The NHMRC, I guess, is Australia's version of the MRC. And the other group of interest that I'm an investigator with is the Australian Trials Methodology Network, or AUSTRIM, who are also a national group funded by the NHMRC in doing methodological research. So today I'm talking about a very recent paper that's just come out in the most recent version of statistical science, as David said, estimating treatment effects from adaptive clinical trials. Of course, adaptive clinical trials will be a term that you're familiar with, but it can mean many different things in different contexts. The unifying theme, I guess, is that adaptive trials have design features that change or adapt during the conduct of the study in response to the accumulating data. And in particular, response adaptive designs have adaptations in response to the essentially the participant outcomes. So as those outcomes are accumulating, design features can adapt to that. What sort of features can adapt? I've listed a few here. You can probably think of a few others as well. Sample size is an obvious one, stopping early for benefit, harm, futility, or going a bit further subject to a sample size re-estimation. Randomization ratios, or adaptive randomization, is a common way in which clinical trials can be adaptive, and we have some common ways in which we do that, Bayesian adaptive randomization and others. Multistage studies. So these are studies where we can have potentially multiple stages and potentially stop earlier or keep going into subsequent stages. When we combine that with multiple treatments, then we have what's called the multi-arm, multi-stage design, or MAMS design, which is quite a common adaptive design. Adaptive dose finding is another area where we use the data to determine how we change treatment doses. And then population enrichment, where we actually adapt the individuals that we're sampling from, or the populations that we're sampling from, in order to get subgroups that are perhaps more promising in terms of treatment responses. So as I said, there's a number of different areas where we can be adaptive, and you can probably think of others as well. Okay, so if we think about treatment effect estimation from adaptive studies, and in particular response adaptive studies, typically the approach that would be taken is to perform a standard analysis that ignores the adaptive nature of the design. There are a few reasons for that we're going to touch on throughout the talk, but it does introduce a fundamental statistical issue, which is the fact that the design itself from an adaptive design is essentially random. The realized design that we get when we run an adaptive trial is essentially a random variable. And more than that, it's a random variable that has a distribution that depends on the actual treatment effect. So it's the design that we end up with in the end is actually informative about the treatment effect. And the implication of that is that standard treatment effects from adaptive clinical trials can potentially exhibit bias. And so that's really the fundamental motivation of the work that we're doing here. And it raises the question that in addition to a standard analysis, and I'm not suggesting that we wouldn't perform a standard analysis for an adaptive clinical trial, but in addition to a standard analysis, should we be considering looking at other sorts of analyses and assessing whether or not those standard analyses are potentially biased? Now, when I talk about bias, there's actually two sorts of bias that we need to keep in mind. The first one being unconditional and the second one being conditional. So both forms of bias are essentially a difference between the average treatment effect from the study and the underlying true population effect. But the unconditional version of that bias is essentially averaged over all of the possible study designs that could occur. So we just said that the design that we get from an adaptive clinical trial is essentially a random variable. And so there's various possible designs that we could end up with. The unconditional bias is essentially the bias that's averaged over all of the possible study designs that could occur. Typically, this is actually fairly small. It doesn't have to be zero, and it is worth studying and thinking about, but it does tend to be small, the unconditional bias. And the upshot of that is that meta-analyses of adaptive studies will tend to be less biased. They'll tend to be unbiased because essentially we're getting a sample of all of the possible designs that we could get and then averaging over them. So meta-analyses tend to be unbiased. But for individual studies, we have this conditional bias, which is the average difference between the treatment effect and the population effect, but for the particular study design that did occur. And that can actually be large or small, depending on the particular study design that we ended up with. So we can have individual adaptive studies that lead to unbiased treatment effect estimation, but also quite substantial underestimation or even overestimation. And this has been studied previously in the context of group sequential studies. And I'm just going to take you through a few examples of this bias here, beginning with this more traditional manifestation of it in the context of traditional group sequential studies. So on the slide here, we have a traditional O'Brien-Fleming stopping boundary for a group sequential study. I've lifted this plot from the Jennison and Turnbull book on group sequential methods. And we see the typical sort of stopping boundaries. We have a standardized treatment effect that we monitor over the course of the study. If it crosses the boundary, then we will stop early. Otherwise, we'll continue through to the planned sample size. In this example, there are 5 analyses, 4 interim analyses, and 1 final analysis. And so in this context, the random design element of this adaptive design is the random sample size. The sample size is not fixed, it's a random variable, and it's a random variable that depends on the treatment effect. So if the treatment effect's larger, for example, we're more likely to get a smaller sample size. And then one of the early studies of this conditional bias, and there was a few papers in the late 90s and early 2000s, this one from Van de Metz and Lan, studied the conditional bias essentially in that O'Brien-Fleming stopping boundary. So this is a simulation study from that paper. We've got the 5 interim analyses and then an overall average treatment effect as well. It doesn't really matter what sort of treatment effect measure we're using here. You can think of it as a log odds ratio or log relative risk or whatever. But the point is that for studies that stopped early at the first interim analysis, we get a very large bias, and the 3 columns in each case are just the different size treatment effects. So you can think of this as a small, moderate, and large treatment effect. In each case, when we stop early, we get a very large bias. So that's essentially conditioning on stopping at the first analysis, we get a large overestimation. Conditioning on stopping at the final analysis, we actually get underestimation. And when we average it over all of the studies, regardless of when they stopped, we get very close to unbiased estimation. So the unconditional bias is small here, but for a given stopping stage, depending on which particular realized design we had, we can have very large bias. And so the question is whether or not we can take that into account in our treatment effect estimation. Another example is adaptive randomization, of which there are many flavors, if you like. The simulation studies I'm going to show you are based on a type called randomized play the winner, which is one of the early ones. To be honest, if you were going to do adaptive randomization in a real study, you probably wouldn't use randomized play the winner, but it's a useful construct to use in the simulation studies, particularly as it has theoretical properties that are well known. And basically the idea is essentially to progressively favor the treatment arm that is doing better. And we do that effectively by, for the two treatment groups, the experimental treatment T and the control treatment C, we essentially add a ball to an urn or a box whenever we get an outcome that favors the experimental treatment, we add a treatment ball. And whenever we get an outcome that favors a control treatment, we add a control ball. And so effectively over time, the treatment arm that's doing better will end up with more balls in the box and be favoured as part of that randomisation process. In this case, the random design refers to the random allocation proportion. So at the end of the study, we'll have 61% in one arm and 39% in the other, and that's the realisation of a random variable essentially. And here is an example of that particular random variable. So this is the final allocation proportion to the experimental group in a simulation of the randomised play the winner adaptive randomisation. Underlying this is a response probability of 50% in the experimental treatment and 25% in the control. We know theoretically that means that the average experimental allocation to the experimental treatment will be 60%. That's a theoretical result we won't go into here. But what happens is that in practice, the actual allocation will vary around that 60%. And that's essentially the realisation of our random design. Now, what happens to the treatment effect estimate is that, and here I'm simulating log relative risks, essentially. What we see is that for designs that fall in this typical range around the mean allocation proportion of 60%, we get very little bias. But when we get extreme designs where the allocation falls quite a way away from the typical value, then we get quite substantial bias, either in the positive or negative direction. That's an example of that conditional bias that we saw with the O'Brien-Fleming stopping boundary. And finally, in terms of examples, I'll just do this third one before we get into some of the concepts. This is just a single arm multi-stage study, essentially a generalization of a Simon type design, where we're studying the proportion of patients that respond in this single arm in 3 stages, different sample sizes in each stage. And we continue if we fall within this region between these 2 boundaries, and we progress to phase 3 if we go above it and discontinue if we go below it. And we can then look at how that simple standard estimate of the proportion responding, which is just a simple proportion, how that responds conditional on the particular design that's realised. This is a simulation study of that design where we look at just studies that made it all the way through to the 3rd stage. The simulations are based on different underlying true response probabilities. And we see that this can either be unbiased depending on what the response probability is, or quite biased for other values of that response probability. So depending on what the underlying parameter value is, you can get quite large bias or potentially no bias in a conditional sense for that multi-stage design. Okay, so let's think about then the data that we get from an adaptive design or an adaptive trial. It's actually, for those sort of comfortable and familiar with stochastic process type context, the data that we get from an adaptive trial is actually a stochastic process. I won't sort of emphasize that characterization here, but that does underlie a lot of what I'm about to say. But we have a sample of observations from an adaptive trial with sample size n. And one of the key defining features is that each observation involves both an outcome, which we'll refer to as X, and this might be the response to the treatment. So did the patient respond or not respond? In everything I'm going to discuss, I'm going to restrict to the situation of an immediate binary response, and that's a whole another area, accounting for delayed responses and that sort of thing. But to keep it simple at the moment, we'll be talking about an immediate binary response. And that's the first element of the observation. And the second element is a design feature, which is allocated adaptively in an adaptive design. And the easiest way to think about that is that it's the treatment allocation, but it could be more general than that, a continuous dosage variable or whatever. And when you see the Y value without the script notation, then we're talking about an individual observation. When you see it with a script notation, we're talking about the whole history of the observations up until a particular observation value. So script YT would be all the observations up to the teeth observation. And I referred to a sample size n. That can actually be random as well. Again, drawing on that stochastic process characterization, n is allowed to be at what we call a stopping time with respect to that particular stochastic process history. Now, one thing I'm suppressing here that if you're interested, you can look at in the paper, we can have a third component to the observation, which is a covariate. And as I said, I'm going to, just to keep it simple here, I'm going to suppress that, but there are also some interesting elements involved in taking account of that, and I will, when we come to talk about the assumptions, just make a couple of comments about that component as well. So that's the data that we get, a sequence of observations that have two components to them, an outcome and a design feature, such as a treatment allocation. There are then two distributions that are of particular importance in defining how that data are generated. The first one is what we'll call the outcome distribution, which is the distribution of the outcome response given the design feature, such as the treatment. And that determines the manner in which the outcomes depend on the design features, and is usually the thing that we're most interested in. And it'll depend on a parameter of interest, which we'll call the treatment effect. But there's also another distribution, which we'll call the adaptation distribution, which is essentially governing the distribution of the design feature, given the history of the observations up until just before that design feature is allocated. And this determines the manner in which the experimental design adapts to the accumulating data. Generally, this will be a distribution. In theory, it can be degenerate if there's no randomness in the way we allocate the treatment arm. So, for example, the original Play the Winner, the Marvin Zellen version of Play the Winner was deterministic, but generally speaking, this will be a distribution, and DT will be a random variable, and it determines the distribution of that design feature allocation conditional on the observation history up to that point. So those two distributions are the key distributions. Just to for concreteness, if we look at what they would be in one of our examples, the randomised play the winner adaptive randomisation. The outcome distribution is pretty simple, just a Bernoulli distribution, probability of response, depending on which treatment arm the person or the participant is allocated to. The adaptation distribution in this case depends on that EARN model that I described, and it can be written down. We don't really need to unpack exactly what all this notation means, but it basically reflects the fact that the probability distribution of the treatment allocation will depend on the number of successes and failures that we've had in the previous observations. An interesting thing to note here is that this adaptation distribution doesn't depend on the treatment effect, and we'll come back to that in a minute. But they're just they're just an example of those two distributions within one of our examples. All right, so before I get to thinking about how we might estimate the treatment effect adjusting for this conditional bias and the adaptive design that we talked about earlier, I want to talk about two basic assumptions that we're going to make. The first one is just an independent sampling assumption, or in fact a conditional independence, which says that the distribution of the outcome given the design feature and the history of the process is actually not dependent on the history of the process. So the outcome is independent of all of the previous observations once I know which design feature was allocated. So essentially, the only information that comes from this process history, or the observations that occurred before the particular observation that we're discussing, the only information that comes from that is contained within the design feature. There's no additional information contained within that process history. It's conditionally independent, essentially. And you could probably see that that's going to be satisfied for many designs. It doesn't exclude things like cluster design. So X here could be multivariate. So it could include clusters of correlated observations, as long as those clusters have the same design feature. And of course, it would also include individually randomized studies where you're assuming independence between individuals. The other assumption, which is really the sort of key assumption, is what I call non-informative adaptation, which is that the design feature does not contain any information about theta conditional on the prior observations. So the design information marginally must contain information about theta because it's adapting to the data, which contains information about theta. But once we condition on the prior observations up until this point in time, then the design feature distribution does not, the adaptation distribution, as I've called it, does not contain any information about theta. All of the information in the design feature comes from that previous observed data that we're adapting to. And we refer to that as non-informative adaptation. Now, the reason why we make those two assumptions is that it then essentially gives us this property of what we'll call the unconditional likelihood function, which is essentially the likelihood function associated with the combined information contained within the design and within the outcomes. And it gives us a result that many of you perhaps be familiar with in the context of sequential sampling, where we know that the likelihood function associated with a sequential sampling scheme is actually the same as the likelihood function associated with a comparable fixed sampling scheme. So the sampling scheme itself is actually irrelevant to the likelihood function. If we make these two assumptions, independent sampling and non-informative adaptation, then that result essentially generalizes to adaptive designs as well. And we can see that here So I'm writing down here the likelihood function of the sample that I described a couple of slides ago, the observation YT, which is the bivariate XT and DT, and this is the likelihood function associated with that sample, YT, given the history of the process up to the time t multiplied over all observations. Now, this is the bivariate XT and DT, so we can split that out into XT given DT multiplied by DT, or conditioning on the history of the process. And what we see here is that our two assumptions essentially allow us to reduce down to this term here, which is the likelihood function associated with a fixed design where we allocated those design features in a non-adaptive way. And we can see that because this here is the adaptation distribution that I talked about, and non-informative adaptation was the assumption that this doesn't depend on theta. And so that term drops out and the equal sign is replaced by a proportional sign. And then here is our conditional independence assumption, where this term becomes independent of the process history. So our two assumptions effectively allow that unconditional likelihood to reduce down to the likelihood associated with a fixed design study. And that's really the motivation for a standard analysis, just using the unconditional maximum likelihood estimator, for example, when applied to an adaptive design. So the fact that the likelihood from an adaptive design is the same as the likelihood from an equivalent fixed design provides a strong basis for just doing a standard treatment effect estimate, or just using a standard treatment effect estimate. But on the other hand, we've also seen that that can give us quite strong conditional bias. So for particular realizations of the adaptive design, we can get quite biased results. And so that motivates the question of whether there are alternatives that we should be looking at, which perhaps adjust for that conditional bias, and also begs the question how those alternatives might compare with the standard unconditional estimate. And that brings us to the idea of conditional estimation, and in particular, conditional estimation of the treatment effect conditioning on the particular design that we happen to get in our adaptive clinical trial. So let's think about what conditioning does in this particular situation. Conditioning on the realised design essentially interprets the observed treatment effect using knowledge of the specific design that did occur, rather than with reference to all the designs that could have occurred. So it really makes the inference more relevant, more specific to the particular design that we ended up with, rather than interpreting it with reference to all of the designs that could have occurred. And that's a feature of conditional inference more generally, that it makes the inference more relevant, essentially, to the particular design that we have. The fact that a particular design configuration has occurred, so for example, the fact that we stopped early due to a large treatment effect in a group sequential study, contains information about how we expect that treatment effect estimate to behave. And so, for example, in that case, we know that we therefore expect it to be an overestimate. And so we can therefore use that information to essentially adjust for the particular design that has occurred. And operationally, the way that we do that is via conditional maximum likelihood estimation. And the conditioning here, as I said, is conditioning on the realised design. Now, before I actually describe the conditional maximum likelihood estimation approach, I actually want to cut to the chase and show you some results associated with those three examples. And then we'll come back and actually describe the mechanics behind the conditional maximum likelihood estimation. So I'm sort of giving you the answer before the question here, but I thought it'd be useful just to begin by illustrating the results with reference to the particular examples that we talked about earlier. So the grey here is the results for the randomised play the winner simulations. In fact, I'll just concentrate on the adaptive randomisation example. And I showed you that conditional bias that exists with the standard estimate, the unconditional MLE essentially. When we use a conditional maximum likelihood estimate in that scenario, you see that that conditional bias actually disappears. So not only do we have unbiased estimation for the typical design realizations, we also have unbiased estimation for the atypical extreme design realizations. So that looks really good, really. And same thing, yeah, I do have the third example here as well. So the multi-stage, three-stage, one-arm study. The grey here is the potential bias that we get for different values of the response proportion. When we apply conditional maximum likelihood estimation, that bias essentially disappears. So looking at those two plots, we might say, well, that looks fantastic. Conditional maximum likelihood estimation seems to get rid of that conditional bias very well. But the problem, of course, is the loss of statistical information. So when we condition on the realised design, we're actually conditioning on something that contains information about the treatment effect. And we're therefore losing whatever information was contained in that particular statistic, if you like, thinking of it like a statistic. And for those of you familiar with the concept of ancillary statistics, the realised design is not an ancillary statistic. It's not a statistic that has a distribution independent of theta, the treatment effect. So we lose information, and you can see that illustrated here. This is the efficiency of the conditional approach relative to the unconditional approach for the adaptive randomisation example. For the very typical design realizations, the conditional approach is quite inefficient compared to the unconditional approach. Okay, so the efficiency is down as low as 50% almost. For the atypical realizations of the design, however, we're getting quite strong improvements in efficiency by using the conditional approach. So the point is that by using a conditional approach, we can get rid of the bias, but we may also be sacrificing information. And so we need to think about balancing those two competing principles, essentially. Okay, so let's, I told you I'll give you the results first, but let's go back to actually talking about how this conditional maximum likelihood approach to estimation would be defined. It requires us to think about two additional sort of characterizations of the information through a likelihood function. One related to the design information and the other related to the outcome information. And that second one will be the conditional likelihood. So we need to think about the information that is provided by this random realization of the design. And to think about that, imagine that we reduced our sample down to just the design information, which we'll call the realized design. And it's essentially the elements of the observations that relate to the design features, but omitting all the information related to the outcomes, the X's. Now that is a random variable that has a distribution, or it's a sample essentially, that has a distribution that depends on the underlying treatment effect. It's informative, therefore, about theta, and it has an underlying likelihood function that we can refer to as the design likelihood. It's actually potentially extremely complicated because it involves integrating out essentially the X's, the outcomes in the overall likelihood function. But in principle, it's well defined. And we can think of it as the likelihood function associated with only observing these adaptively assigned design features. In principle, then, we can actually use that to estimate the treatment effect theta without the outcome information. And that then leads us to the conditional likelihood, which is just the ratio of the unconditional likelihood function that we had earlier, that I showed was insensitive to the adaptive sampling scheme, and divided by that design likelihood that I referred to earlier. Or if we look at it on the log scale, it's a difference. And that then leads, as I said, to, well, actually 3 different versions of what we might call the maximum likelihood estimator. The conditional MLE, which is based on the conditional likelihood, the unconditional MLE, which is just the standard MLE that we talked about earlier, and what we'll call the design MLE, which is just the maximum of the design likelihood function or the associated score function. So I've got 3 different estimates here, all of which are based on different elements of information contained in the sample: the design information, the outcomes given the design, and the overall information combining both components. And that then leads to what I think is a fairly important concept within the context of adaptive designs, which is something that I'll refer to as the design information fraction. So this script F, which is essentially the proportion of the overall information about the treatment effect that is provided by the observed realized design that we happen to get. And so this design information fraction is essentially the ratio of the information that comes from the design likelihood and the information that comes from the overall unconditional likelihood. And it'll be necessarily less than 1, between 0 and 1. And it quantifies essentially how much of the total information is coming from the adaptive design. So for example, the final allocation proportions in an adaptive randomization design, how much information about theta is coming from that particular observation. Underlying this information definition is just the sort of traditional observed information associated with each of those 3 different likelihood functions. So the unconditional, the conditional, and the design likelihood. And this equation here is essentially the decomposition of that overall information in the entire sample into the sum of the design information and the outcome information conditional on the design. And so the design information fraction is just re-expressing that decomposition. Now, the reason why the design information fraction is a useful concept is that it gives a useful way of thinking about the relationship between these 3 types of information, or 3 types of treatment effect estimates. What we saw was that the unconditional likelihood is essentially a product of the conditional likelihood and the design likelihood. That product can essentially be interpreted as an information aggregation of 2 independent sources of information. The source of information coming from the design and the source of information coming from the outcomes conditional on the design. And if we adopt some first order approximations to those likelihood equations that I had on previous slides, we can actually write down a very interesting approximate representation or characterization of the standard MLE, the unconditional MLE, as a weighted average of the conditional MLE and the design MLE, where the weighting is determined by the design information fraction. So it's sort of a meta-analytic analogy. The overall information in the sample is essentially a meta-analysis of the design information and the outcome information given the design, weighted according to this design information fraction, which is it's a helpful way of thinking about that decomposition of information. And it also leads to some natural ways to kind of explore the potential for bias in that unconditional approach. Okay, so we've got this dichotomy or competing concept, if you like, where the conditional inference approach seems to lead to unbiased estimation. Certainly in those examples, it was very effective at providing unbiased estimation conditionally, but it can involve a loss of information. Whereas the unconditional approach may lead to conditionally biased estimation, but overall will often be more efficient than a conditional approach. And so the question is, can we examine the presence of conditional bias and information loss as part of the analysis process? So within a given study or within a given experiment, can we actually explore the extent to which we've got conditional bias versus information loss, and actually then make some assessments about which approach, conditional or unconditional, would be better for estimating the treatment effect? And the answer, I think, is yes. And the key consideration here is the idea or the question of how extreme is the particular study design that we have observed in our particular study. And we'll see that this notion of extremity of the study design plays a key role in determining the magnitude of the conditional bias. Related to some of those identities that I wrote down on the previous slides, another key relationship that we can write down relating those three estimates, the unconditional MLE, the design MLE, and then also in the background is the conditional MLE. What that leads to is the following relationship with between their respective expectations and then hence their respective biases. And the key sort of relationship here is that the conditional bias is equal to the unconditional bias plus a measure of the extent to which the design is extreme. And by that, I mean the difference between the design MLE and its expectation. So if we've got a design that is quite typical, then this term will be 0. If we've got a design that is quite extreme and unusual relative to what we would expect on average, then this will be non-zero. And that's what I'm referring to as the design extremity. So if we have an outlier, then this conditional bias will tend to depart a great deal from the unconditional bias, bearing in mind that typically the unconditional bias is very small. So we would typically have a large conditional bias for an outlier design, but not much conditional bias for a typical design. And it then begs the question, can we actually determine whether our particular study is at risk of having this extreme design? Because one problem that you see here is that this measure of the design extremity depends on theta, the unknown theta. So I can't actually calculate that for an observed study. I can calculate it in my simulation studies where I know the true theta, but I can't do it for a real study. So that begs the question then, can we use that in practice? And the answer is yes, using essentially a surrogate for that design extremity, which is the heterogeneity or just the difference between the design information and the outcome information conditional on that design information. And so what I'm going to point to here is two measures, one of which is observable. The ZH is what I'll call the heterogeneity statistic. It's simply a standardized version of the difference between the design estimate and the conditional estimate. And then the design extremity value, which is based on this unobservable quantity that depends on the unknown theta. And I'll appeal to a result that says that those two quantities have a certain correlation that depends on the design information fraction. And typically in these sorts of adaptive designs, you're not going to expect the design component of the information to be the majority information. In fact, typically you wouldn't see that design information fraction exceeding, for example, 1/3. So this correlation is going to typically be 80% or more in real studies, one might expect. So there's a decent level of correlation there such that we can use this observable surrogate for the unobservable measure of the design extremity. And I've just illustrated that here in the bottom right-hand corner. So this is now some more simulation results for our adaptive randomization example. And you see that in those simulations, plotting the heterogeneity Z versus the design, unobservable design Z, it's unobservable in practice. Of course, in my simulations, I can observe it. And we see that strong association such that we might use that as a measure of the extent to which the design is extreme. And what we also see up here is a representation of the behavior of the different treatment effect estimates for different realized designs. So you remember this, this design had an average allocation proportion of 60% in the experimental treatment arm. And in that case, we get on average very similar conditional and design estimates, and hence unconditional estimates. Whereas at the two extremes, where we have less than the typical value, 55%, or greater than the typical value, 65%, we get some discrepancy emerging in opposite directions between the conditional and the design information. Now, these are simulation results, but they do sort of suggest the usefulness of a forest plot type of presentation. And that's what I've done here with our multi-stage study. So this is now not simulation results. This is, I've already showed you the top left panel here, so you can ignore that one. But the other panels represent two essentially samples that we could get. So the top panel here and the bottom panel, and then their associated likelihood functions, respectively. In both of these samples, as with the simulation results I showed you earlier, we're looking at multi-stage studies that made it all the way through to the end. And so that is reflected in the design estimate and its associated confidence interval. But the actual proportion of successes or responses differs substantially between the two samples. So in this case, we've got a conditional maximum likelihood estimate that is quite consistent with the design estimate and therefore consistent with the standard unconditional MLE. And in that case, I have a Z statistic measuring heterogeneity that that is very small and you would interpret these on a standard normal scale, essentially. Whereas in the second sample, we see that discrepancy emerging, and it is diagnosed by that Z heterogeneity statistic. And so in this situation, you would probably draw into question the unconditional estimate and consider using the conditional estimate that essentially conditions on the design, because that is the situation where in the context of an extreme design, the conditional approach gives you more unbiased estimation. Now, I'll just briefly mention that that approach is essentially about diagnosing bias or using a diagnostic for bias. The other approach is to say, well, let's modify our estimation procedure so that it can sort of dial its way between the unconditional and the conditional approaches, such that when the unconditional approach is more appropriate, it goes more in that direction. When the conditional approach is more appropriate, it goes in the conditional direction, and just let the sample decide how far in either direction we should go. And that's achieved using this concept of a penalized likelihood approach. So the unconditional likelihood is on the log scale, log likelihood, it's the sum of the conditional and design log likelihoods. If I add in a tuning parameter here, well, these are the score functions actually. If I add in a tuning parameter, then I essentially generalize that notion such that for the tuning parameter being 0 or 1, I recover the conditional or unconditional likelihoods respectively. But for values in between, I can sort of dial it from one extreme to the other and let the sample decide which might be more appropriate. I won't go into all the details. Has some concepts related to robust statistics as well there, but I'll just show you some results of that. So the top two panels you've effectively already seen. This is the unconditional and the conditional for this adaptive randomization scenario. When we use the penalized likelihood approach, we get relatively unbiased estimation. But what you see here with the efficiency is some important advantages. So the scale on which this is measured, values greater than 1 mean that the estimate is less efficient than the standard unconditional MLE. So as we said earlier, the conditional MLE can be quite inefficient for typical realizations of the design and more efficient for atypical realizations. The penalized MLE retains that better efficiency for atypical realizations, but isn't so susceptible to a loss of efficiency for the typical realizations. And that, again, as I said, has some elements that are analogous to robust statistics as well. Now, I'll just, before I put up my last slide, which is my conclusion slide, I'll just answer this question. What would an atypical design look like in practice? Can I give you an example of one? This is not an adaptive design, but this is an example of a study which is used to illustrate how extreme sort of paths you can get as you monitor a study. This is the ISIS-1 study. And you see that what happened was that the treatment effect went very strongly in favour of placebo before reaching significance in favour of the experimental treatment, atenolol. If we were to have done adaptive randomisation there, although there's a strong treatment effect, the adaptive randomisation would have favoured placebo early on and probably taken some time to catch up. And ultimately, we would have had an allocation proportion that was somewhat out of whack with the fact that there is a strong treatment effect there. So depending on the path that the study takes, that realised design could be quite extreme. So in conclusion then, this is the last slide, the primary treatment effect estimate will be the typical standard analysis. But what I'm suggesting here is that we have a supplementary analysis based on this conditional maximum likelihood estimate. And it's the heterogeneity between the design information and the outcome information, or equivalently the extremity of the design, that really gives us information about whether that conditional approach is likely to be less biased than the unconditional approach and the associated visual representations of that as well. I think I'll leave it there. Thanks very much.
Great, thanks very much, Ian, for a really interesting, clear talk. I'd like to open the floor up for questions. So I think you can raise your hand if you go to the bottom of the screen onto reactions, or feel free to type in the chat. So maybe I'll start while people are doing that. So I guess I wonder in kind of practice, you could see you might have a unconditional estimator, a condition estimator, and then this kind of penalized estimator. So would you suggest in practice that all three are presented, or just the kind of forest plot that you were showing earlier?
Look, I think, so at the moment, I'm not sort of, it has been a basic principle that we do a standard analysis of adaptive trials. So I'm not sort of suggesting that we turn that on its head. But I think that as a supplementary analysis, presenting the forest plot representation of the three estimates, and then potentially also the penalized estimate, which is then essentially a fourth estimate, just gives us information about whether that particular realised design is susceptible to conditional bias. So it's, I'm not sort of saying replace the typical approach, but use it as essentially a way to explore the susceptibility to conditional bias.
Great, thanks. Sean, I think your hand was up first.
Yes, thank you very much. That was very interesting. You presented the penalized estimator as a compromise between bias and efficiency, and I was just wondering whether you were balancing those in some formal sense, like trying to minimize the sum of squares, mean of sum of squares, or something.
Well, what I was, and I must admit, I accelerated a little during that slide, so I might have gone over it a bit quick, but I made that tuning parameter a function of the measure of the design extremity. So when the design is atypical, that tuning parameter would become 0 or close to 0, which means it's moving towards the conditional MLE. When the design is typical, that tuning parameter would go close to 1, which then makes it close to the unconditional MLE. So using that, the measure of design extremity to kind of control how far you go in each direction.
But how do you, how do you decide what lambda is? I understand what you've just said, but are you choosing lambda in order to minimize some objective function?
So in fact, lambda is a function of the treatment effect theta. So it just becomes another place in the likelihood equation or the estimating equation where the treatment parameter theta occurs. So you're actually solving an estimating equation in which theta is occurring in an additional place within that estimating equation. So I'm not actually choosing lambda and then putting it in. I'm actually letting the whole estimation process determine what that value of lambda is by virtue of lambda's dependence on theta.
Right, but
it's what you might call adaptive, and they use the word adaptive in a different way in penalized likelihood estimation, but it's adaptive in that sense.
Okay, thank you.
Yeah.
Thanks, Jack.
Thanks, Ian. Brilliant talk. I've heard the argument that every design is adaptive because some changes invariably are going to happen from starting the study to the end. So much of what you've talked about depends on pre-planned design adaptations and how much could be sort of not pre-planned, because at the end of the study, you know what has happened already anyway.
Yes, look, how much is determined on pre-specification of the adaptation? Pretty much everything that I said is dependent on that, in fact. And I probably should have put in my definition of an adaptive clinical trial that the adaptations need to be pre-specified, at least in the way that I'm studying them. So that adaptation distribution is based on the pre-specification of how we will adapt to any given observations that occur. The unplanned adaptations that you're referring to would not be accounted for in this particular context, because we need to have an underlying adaptation distribution. The way in which we adapt, even if it has some randomness in it, needs to be essentially pre-specified in this framework.
Okay, and Sophia.
Thank you, Ian. This was a really nice talk. I enjoyed it a lot. I had a question around what were your thoughts? Is there a consequence for design of all of this? Because when, especially for response adaptive randomization, would it make sense to choose a procedure that has less chances of extremity or less of a variance in its kind of extreme designs? And would that make sense? Particularly when you said we wouldn't use randomized play the winner in practice. I think it's probably that would explain it. And yeah, I was wondering what were your thoughts around that?
Yeah, that's a really good point. I think that could be one consequence of this, that you could favour. So, for example, in that adaptive randomisation example, you could favour schemes that, we already favour schemes that have less variability, but this would be another way of looking at the advantage of those schemes. And my comment about that we wouldn't use randomised play the winner was exactly what you're referring to, that there are always other schemes that for the same kind of target have smaller variability. So this would essentially perhaps offer an additional advantage of those reduced variability schemes.
Okay, great. So I can't see any other hands raised or comments in the chat. So, and we're a bit over time, so I'd just like to thank Ian again for a really great talk, really interesting work, and I'm sure we can all join in thanking him for the talk.
Thank you, and thank you as well, David, and everyone that came. I enjoyed it, and as I said, I hope that we can find ways to continue to interact. I think we're doing similar stuff and working on similar areas, and I hope that we can, as I said, interact in the future. Thank you.
That'd be great. Thanks.
Thank you.